Vitamin B9, or folate, is an essential water-soluble molecule. While it is required for human survival, the body cannot synthesize it and must obtain it from external sources. Total body stores are relatively small, ranging from 0.5 to 20 milligrams. Without regular dietary intake, these reserves can be depleted in as little as 3 weeks or up to 6 months. We acquire this vitamin through dietary sources, like dark leafy greens, nuts and meats, and fortified grains and cereals. The form of the vitamin dictates its bioavailability. We only absorb about 50% of the folate found in food, whereas synthetic folic acid supplements offer 100% absorption due to their molecular structure. Once ingested, folate travels through the stomach and enters the small intestine for extraction. Absorption occurs specifically in the proximal duodenum. The medical mnemonic F is used to remember that folate, iron, and calcium are all absorbed in this initial section of the small intestine. After entering the bloodstream, the vitamin is processed by the liver. Because it is water-soluble, any excess that the body does not immediately use or store is excreted through the urine. Because the body flushes excess folate and only extracts half the volume in natural food, a consistent daily supply is necessary to maintain baseline stores. To understand why the body has this daily requirement, we must look at the biochemical reactions occurring inside the cell. As raw folate enters the cell, it is immediately acted upon by the enzyme dihydrofolate reductase, or DHFR. DHFR converts folate into dihydrofolate and then processes it a second time to create the master active form, tetrahydrofolate, or THF. THF is then processed by other enzymes to form a specific intermediate called N5-N10-methylene THF. In the first major utilization path, the enzyme thymidylate synthase uses this intermediate to convert DUMP into DTMP. This conversion is the essential step for pyrimidine synthesis, which provides the building blocks for DNA strands. Simultaneously, a parallel branch generates N-formal THF, which is required for purine synthesis. Without this enzymatic cascade, a cell cannot replicate its DNA, which makes folate availability a primary requirement for cellular division. When the N5-N10-methylene THF intermediate is not used for DNA synthesis, it can be diverted into a secondary metabolic path. It is converted into 5-methyl-THF, which enters a mechanism known as the activated methyl cycle. Within this cycle, 5-methyl-THF pairs with vitamin B12. The enzyme methionine synthase then facilitates the transfer of a methyl group to homocysteine to create methionine. This methionine is subsequently converted into S-adenosyl methionine, or SAM. SAM serves as the necessary fuel for the synthesis of nucleoproteins and catecholamine neurotransmitters. These metabolic pathways also provide a clinical solution for methanol toxicity. Ingested methanol breaks down into a toxic byproduct called formic acid. High doses of folate can be administered to increase the rate at which the body metabolizes and clears this toxin. Folate acts as both a structural component for DNA and a driver for the chemistry behind neurotransmitters and toxin clearance. These cellular requirements are the basis for the specific dietary guidelines established for the human population. The recommended daily allowance scales as a patient ages. For children between 1 and 3 years old, the RDA is 150 micrograms. This requirement increases to 200 micrograms daily for children ages 4 to 8. For those between the ages of 9 to 13, the daily requirement rises to 300 micrograms. For everyone over 13, the standard adult maintenance dose is 400 micrograms per day. This 400 microgram baseline represents the amount needed to support cellular turnover in a healthy adult. Standard adult requirements are superseded during 2 specific physiological events: pregnancy and lactation. Lactation increases the metabolic demand on the mother, requiring an intake of 500 micrograms per day until breastfeeding is complete. During a normal pregnancy, the rapid division of fetal cells increases the maternal requirement to 600 micrograms per day. However, the risk of neural tube defects may necessitate higher doses. High-risk factors include previous NTDs or the use of anticonvulsant medications. In these high-risk cases, the dosage escalates tenfold to 4,000 micrograms per day. This high-dose protocol must begin 1 to 3 months before conception to ensure adequate levels are present from the start. This level of supplementation is maintained through the first 12 weeks of gestation, covering the period when the embryonic neural tube physically closes. This tenfold increase in intake supports the surge in thymolate synthase activity required to replicate the DNA needed to build a human nervous system.